Good afternoon, everybody. Thanks for coming, and we have a very special presentation today, something very timely. So for those of you who don't know me, I'm sure everybody knows me by now, right? But anyways, just in case, I'm Ethel Kagan. I'm the AD in the IDGI pulmonary group, and I think a lot of you know that the GI group, to a great extent, or to big extent, huge extent, is based on Dr. John McCutcheon coming to the DCRI and really creating around hepatitis C a very active and very large clinical research group. And certainly Hep C is a very important disease in the community, and for those of you who worked in IDGI know that a lot of our trials are in Hep C, and with some of the discoveries that have been made that actually Dr. Alex Thompson was involved with, and I guess, Alex, you're going to talk a bit about that as well. Hepatitis C is even more interesting out there in the community, and we're certainly getting a lot of contacts by other companies to come and do more Hep C trials. So this is a very important disease entity for the DCRI, and I don't know if everybody realizes that while the cardiovascular group is the 800-pound gorilla at the DCRI, actually GI is actually the second largest group. The IDGI pulmonary group, we're actually the second largest group. And recently, when some people from the C4 were presenting some data, we really represent about close to 25% of actually the research dollars that come into the DCRI. So thanks again for coming. I know Friday afternoon, everybody's very busy, and so I really appreciate your attendance. So Dr. Alexander Thompson is actually a physician PhD, and he is a postdoctoral research fellow who comes all the way from Australia, and we're really very lucky to have him here today. And he has agreed kindly that if you have questions during the presentation, he's happy to answer them. There are mics and around the room. So without further ado, Dr. Thompson.
Thank you. That's great. I wanted to start by just echoing Ethel and saying that the liver disease is an important problem. We can't compete with the cardiologists. You can see this slide summarises the top 10 causes of death worldwide, and you can see that ischemic heart disease is number 1. But liver disease is on the list. That's number 7. And the major causes of death due to liver disease are cirrhosis and cancer, liver cancer or hepatocellular carcinoma. So I wasn't quite sure how familiar you all were with hepatitis C in the liver, so I thought I'd start pretty basic, and then we'll move to a bit more detail as I go through the talk. So this cartoon, I've got a number of different cartoons through the talk just as some light relief. This cartoon just summarises what the liver does. So we all know the liver is an organ. It sits in your tummy, it sits on the right side of your tummy under your diaphragm, and it has a number of different functions. The primary function is to detoxify the blood. So all the nutrients that are absorbed from your gastrointestinal tract are channeled straight to your liver, where the liver filters things, but it also filters the systemic circulation as well. It has important roles in glucose metabolism, in protein metabolism, in fat absorption, and also produces a number of coagulation factors, so it's important in preventing you bleeding. Now, the most common causes of liver disease in North America are, number 1, hepatitis C, which is what we're talking about today. Number 2, fatty liver disease. Fatty liver disease may take over from hepatitis C as the community becomes more and more obese. It's certainly recognised as an epidemic now, but for the moment the statistics show that hepatitis C is number 1. Then there's alcohol, hepatitis B, and then there's many other much rarer causes of liver disease. Today we're focusing on hepatitis C. So some basic facts. And I'm going to apologise, I can't read that television, so my eyesight's just not good enough. So I'm going to have to turn around to the big screens. Hepatitis C, the virus, was first isolated in 1989. Prior to this, it was recognised as an acute cause of hepatitis in cases where hepatitis A or hepatitis B couldn't be identified. So for a long time it was known as non-A, non-B hepatitis. Once it had been identified, it became hepatitis C. 4 to 5 million people in the US are antibody positive for hepatitis C, and about 3/4 of them will actually have ongoing active hepatitis C. The highest prevalence is among African Americans, males, and subjects between the ages of 40 to 49. As a question, what are the 2 commonest causes of death in a 40-year-old African American male in Harlem? Number 1, a gunshot wound, but number 2, liver disease due to hepatitis C. So this is a big problem in North America. It's acquired through exposure to contaminated blood, most commonly through injecting drug use in this country. Concerning to hepatologists is that 2/3 of patients haven't been identified by medical care, and then of the 1/3 who are under the umbrella of medical care, only 1/3 of them, so a third of a third, are actually treated. As such, hepatitis C is the most common cause of cirrhosis, and it's also the most common indication for liver transplantation. Beyond North America, it's a global problem. So 3% of the world's population, or approximately 170 million people, are thought to be carriers of hepatitis C. 4 to 5 million in the US. It's concentrated in Southeast Asia and Africa, though, where there are greater numbers. In Australia, where I'm from, there's 200,000. We have a population of just over 20 million. So it's about 1% of our population. So what is hepatitis C? Hepatitis C can be thought of as a rude house guest in the liver. So it moves in, doesn't pay rent, doesn't do any maintenance, has lots of kids, and over time it damages the place. And that's what leads to the problems. Now, I don't want to get too technical, but I thought it would be useful if we had some discussion of the virus itself. So it's an RNA virus. On the left here, you can see the RNA genome of the virus, which is surrounded by what's called the capsule, and then the envelope. And this is what circulates in the blood. There are different types of hepatitis C, which becomes particularly important when down the track I'm going to speak about treatment. But there are at least 6 different types of hepatitis C, and they're different because of variation in the RNA genome. So once there's greater than 30% difference between 2 genomes, they're said to be 2 different genotypes. And this figure summarises how different the different genotypes are. And then worldwide, this is the genotype spread. You can see that in North America, genotype 1 is the most common type of hepatitis C. That's in the red. As we move to Southeast Asia, the non-genotype 1 viruses become more common, and genotype 2 and 3 are more common. Genotype 2 and 3 are also more common in South and North Europe. The green circle represents genotype 4, and this is endemic in Egypt because this relates to vaccination practices almost 50 years ago now, but up to 20% of the Egyptian population have hepatitis C. And this is the most technical slide here, and I've included it because it's relevant to the new treatments that are becoming available. So this is the life cycle of the virus. This cell is a hepatocyte. The virus, and I'm just going to walk through the steps of the viral life cycle. The virus binds to the cell membrane. It enters the cell. The core and the envelope proteins are removed, and the RNA is then free in the cytoplasm. From the RNA, a protein called the HCV polyprotein is translated, and that protein is then cut up, and the different proteins do all the different functions of hepatitis C. It makes a number of different functions that help it live within the cell. One of those proteins is involved in replicating the virus, so it's a negative strand RNA virus. It's reverse transcribed to a positive strand virus, from which the negative strand virus is then transcribed. It then moves to the Golgi apparatus and is the envelope, the core. It's encapsulated with the core. The envelope, it is enveloped, and then it's secreted to the circulation. Sure. Yeah, so interrupt throughout. That's fine.
Working. There we go. Simple question. The translation process here, is this endogenous ribosomal activity?
Yes, it is. That's a good question. It is.
Okay. Thanks.
So how is hepatitis C spread? It's essentially spread by blood-to-blood contact. So this slide summarises the groups within the community who are infected with hepatitis C. So you can see, and this is data from North America, so you can see that 70% of the patients we see have acquired their hepatitis C through injecting drug use, through exposure to contaminated blood through unsafe injecting practices. This data so the risk of sexual transmission is controversial. In this data set, which is published in Hepatology and is recognised by the Liver Association, the risk of sexual transmission is 18%. Now, that varies a lot depending on the sexual practices, and we would normally see patients in the clinic 20 years down the track. So the typical patient we would see, or one of the two typical sorts of patients we would see in the clinic, is a 40 to 50-year-old person who on an insurance test has been found to have hepatitis C. They've not known about it until this blood test, and they acquired it 25 years ago from injecting practices, and they may just have been very short-lived and experimental. But that kind of patient, often they're married, we will test their partner, and almost always they're negative. So for someone in a stable monogamous relationship like that, we wouldn't recommend barrier protection. The risk of transmission to the partner is so low that we wouldn't recommend that. Now, in certain high-risk sexual practices, that's a little bit different. So more traumatic intercourse has a high risk of transmitting hepatitis C. But for monogamous couples in a stable sexual relationship, sexual transmission is very rare and barrier protection is not needed. And then there are smaller numbers of patients who've acquired hepatitis C through medical procedures. It's for older patients, blood transfusions before 1992 carried a very high risk of hepatitis C. And there's a small risk of perinatal transmission from mothers who have active hepatitis C to babies. It's about 5%, maybe a little less.
Alex, can I ask you a question about the perinatal transmission? Mm-hmm. Is that entry in utero, or is that from passing through the canal and being exposed to maternal blood?
That's a good question.
You know what I mean?
Yeah, we don't really know the answer. So okay. I'll finish off by saying it's a really good question, but I can't tell you the exact answer.
Okay.
Yeah. And this slide, particularly with respect to healthcare exposure, describes the relative risks of transmission for parenteral exposure for hepatitis B, for hepatitis C and for HIV, the three common bloodborne viruses that we're concerned about. So hepatitis B, you can see that hep C is most likely to lead to infection from injecting drug use. From occupational use, hepatitis B is much more common. Overall, hepatitis B is much more infective than hepatitis C. In terms of occupational exposure, it's about 10-fold more infective than hepatitis C, which again is 10-fold more infective than HIV. So that sort of puts it in context for when we start to talk about needle stick injuries and the like. And as a result of these different modes of transmission, this is the prevalence within particular groups within the United States. So haemophiliacs who've been exposed continuously and for many years to blood products, haemophiliacs who were having treatment prior to 1992 have a very high risk of hepatitis C, and you can see that the prevalence, it's almost 90%. Injecting drug users, almost all injecting drug users who follow unsafe injecting practices within a year of starting to inject will be hepatitis C positive. And then haemodialysis patients are also at risk. Clients of sexually transmitted disease clinics, and then it falls away. You can see in the general population that's less than 3%. Surgeons through needle stick injuries are the most vulnerable of healthcare workers, and then it's very rare in other situations. So transmission is highly efficient among injecting drug users. It's rapidly acquired after initiation, 4 times more common than HIV amongst the injecting drug using population, and prevalent 60-90% after 5 years. So that's hepatitis C. How does it affect the liver? So the virus replicates in the liver. You can see the viral copy of 9,000. And hepatitis C replicates much more actively than any other virus, HIV or hepatitis B, approximately 10-fold higher in terms of its daily reproduction rate. And injury in the liver occurs as a 3-stage continuous cycle where we have injury, then we have the body's response to injury leading to inflammation, and then we have the body's repair mechanism that causes scar tissue. So first of all, inflammation. You can think of inflammation and repair as an attack on the liver. Inflammation is directed at trying to get the virus out of liver cells. So the virus itself lives within the hepatocytes. And it's controversial as to whether it can live inside any other type of cell. It definitely lives inside the hepatocytes, and in most cases it's not the virus itself that's damaging the liver, it's the body's immune response to try and get rid of the virus. So inflammation is the first step of this. Immune cells infiltrate the liver, they secrete cytokines, and they can directly lyse cells that contain hepatitis C. A little bit of this is not a problem. The liver can regenerate very nicely, but over time damage can build up. And the scar tissue you can think of as the long-term consequence of continuous inflammation. So the liver can cope up to a point, but gradually fibrosis or scar tissue starts to build up, and that's when we start to see serious liver problems. So this is a schematic of a liver lobule, and this is how we classify, for those of you involved in hepatitis C trials, this is how we classify the progression of cirrhosis. Early in disease, there's really no fibrosis. There are a few inflammatory cells within and surrounding the hepatocytes. Gradually, you can... Sorry, the laser pointer may not highlight. But in the centre, the portal tract, which is in the middle of this cube, you can see that there are little tendrils of grey stretching out from it. That represents fibrosis. And there's a few more inflammatory cells. In stage 2, the portal tract is expanding more, and these tendrils are longer. By stage 3, the tendrils actually extend to all the adjacent portal tracts, and you start to see linkage of them, and that creates little nodules in the liver. And stage 4, which is cirrhosis, is when this fibrosis is so advanced that there's complete architectural distortion of the liver. And that's what cirrhosis is. And it's once we get to the point of cirrhosis that the liver stops coping. So up to the point of cirrhosis, the liver works pretty well. And within sort of cirrhotic patients, a proportion of them, the liver function can compensate, but this is when patients start to be at risk of the liver no longer working properly. And this is what a cirrhotic liver actually looks like. This is an explanted liver. So you can see these nodules, which represent clusters of hepatocytes within the boundaries of scar tissue. So if we think about patients with hepatitis C, if we take a typical 100 patients, and this is a 20 to 30 year time course, 20 to 30% of patients who are exposed to hepatitis C will clear it acutely. So not everyone gets chronic hepatitis C, but most patients do, 70 to 80%. Of those 70 to 80%, about 70% will stay clinically stable and histologically stable over a 20-year period, but the risk of cirrhosis is about 20%. And it's this group of patients that we are particularly concerned about. Again, about 3/4 of them will remain clinically stable, but about a quarter of them, over 20 to 30 years, this is the lifetime of the disease, will die from their disease. So not everyone dies from hepatitis C. Most people don't, but a percentage do, and really we can't identify which patients are going to die. Now, of this 4% of patients who die, death is due to the liver no longer working properly, what we call decompensated liver disease, or the development of liver cancer. And it's about half and half. Now, of concern for the community, I guess particularly this is a hot topic at the moment with the sort of healthcare plan in the news, is that of the pool of patients with hepatitis C at the moment, most of them are pretty well. But over the next 20 years, we're going to see a much greater fraction who are progressing to more advanced liver disease with cirrhosis and then decompensation and also liver cancer. So it's estimated that by the year 2018, these data were generated 10 years ago now. So in 10 years from now, the prevalence of cirrhosis in the community is going to go up 63% within the population of hepatitis C patients. The prevalence of decompensated liver disease is going to go up 84%. Hepatocellular carcinoma is going to go up 63%. And liver-related deaths are going to double. And it's not just in the United States. These are worldwide trends for liver cancer related to hepatitis C. And you can see that in the US, in the 90s, it's triple what it was in the 80s, but also in France, Italy and Japan. There's an increasing rate of hepatocellular carcinoma, largely driven by the hepatitis C epidemic. So what can we do for patients who have advanced liver disease, either decompensated liver disease or liver cancers? The curative treatment is a liver transplant. And so this slide describes the number of the number of deceased donor transplants that have been performed per year for the last 20 years to 2006. if I take the number of the year 2000, so almost 5,000 transplants were performed in this year. If we compare this slide against the number of patients who are on the waiting list for a liver transplant, you can see that exponentially the liver transplant waiting list is completely overwhelming the number of patients who actually receive a transplant. So in 2001 there were 20,000 patients on the transplant waiting list, 4 times the number of transplants that were performed. And this will be expensive. So as the projected morbidity and mortality in the community due to hepatitis C increase, so the costs to the community of caring for these patients are going to increase. And just to quote some numbers, it's estimated that over the next 20 years there'll be $11 billion required for direct medical care expenses of patients with hepatitis C, $75 billion in sort of indirect costs related to hepatitis C. And just to put hepatitis C in the context of another common disease, the healthcare costs in 1997 for hepatitis C were the same as for asthma. So again, hepatitis C is a real and common problem.
Alex, what's the success rate of liver transplants in this population?
So we would quote to patients a 5 years post-transplant graft survival rate of 70 to 80%. Does that answer the question? Yeah, so these patients do pretty well. They're not the best group, but they do pretty well. They do particularly well if you can cure the hepatitis C before they have the liver transplant. Right. So recurrent hepatitis C after transplant can be a difficult problem. So this brings us to the treatment for hepatitis C. So the standard of care at present is a 2-drug combination. A drug called interferon, which is a weekly injection. It's called pegylated interferon, or PEG. You might hear people talking about PEG, with some tablets that are taken every day called ribavirin. About half of people are cured permanently on this regimen. Hepatitis C is the only chronic viral infection that we can cure. We can't cure everyone though. So 50%, and that depends on genotype, which we'll get to. And once people are cured, the virus is gone. So we can reassure patients that the virus is gone. Your liver disease, for patients who are cirrhotic, cirrhosis is not going to go away, but for earlier stages of liver disease, their liver function will improve, and they can be reassured that they have a normal life expectancy. The problem with hepatitis C treatment, there are 2 problems. 1, it doesn't work for everyone, but 2, there are lots of side effects, particularly related to the interferon, and I'll talk about them in just a minute. As a result of this, many patients can't tolerate the whole treatment course. So about 30% of patients within treatment trials, which are sort of the best of the best, about 30% of patients will drop out of their treatment. They won't be able to complete the full treatment course. So
how is cure defined?
So cure is defined as detectable RNA within the blood with very sensitive PCR assays. And the studies that have sort of reassured us that patients are cured are followed up out to 10 years now. So the virus was only discovered in 1989. We don't have lifetime follow-up, but to 10 years for people who've been cured, haven't been reinfected, haven't had other risky behaviours, then the likelihood of being HCV positive 10 years later is less than sort of 1 or 2%. And almost certainly that 1 or 2% are related to reacquiring hepatitis C. So as opposed to hepatitis B, once we can't cure hepatitis B, but if the body can spontaneously get rid of it, you can't get it again. Hepatitis C, if the body gets rid of it, or if we cure it through treatment, you can get it again. So this is the progression, the history and the progression of treatment. Success rates for hepatitis C. So interferon was the first drug that was identified to have some efficacy for hepatitis C. When it was first identified as being an effective treatment, the success rate was 6%, which I'm not sure what the statisticians would think about a trial that showed a success rate of 6%, but it was probably a trial based on 20 patients. So I think we can pretty much conclude that interferon by itself, it has an effect, but it's a very small effect. And then it works a bit better if you give it for twice as long, if you give it for a year versus 6 months. If you pegylate it, which involves putting a polyethylene glycol, attaching that to the interferon itself so that it stays within the blood longer and you only need to have it weekly, it increases again. The combination with ribavirin almost doubled the response rates. And then the combination of pegylated interferon with ribavirin leads to success rates between 54 and 61% of patients overall. So what is interferon alpha? the backbone of the treatment. So it's actually a naturally produced cytokine. So your body makes interferon, and it's the body's natural response to viral infections. It's synthetically produced. So here we have, in the bottom left, we have little interferons that the body is making. We have laboratory-produced interferons going into the liver, where they act to get rid of the virus. And the white circles are the polyethylene glycol, the PEG. And how does it work? We don't know exactly how it works, but we know it has a multitude of different effects. We're not sure exactly which one of them is what's the key for hepatitis C, but we know it directly inhibits viral replication. It's able to directly clear cells of the hepatitis C. It also stimulates the immune response. So it stimulates T cells and B cells to have anti-hepatitis C effects. So this slide summarises the number of patients who are cured or not cured by current treatment regimens. And it would be really nice, we've got 50%, and we see a patient in clinic and we quote them a 50% response rate. It would be really nice if we could refine that prediction of their risk. But at the moment, the factors linked to response, whilst we know some of the factors that predict response, our ability to judge exactly who's going to respond and who's not is not very accurate. So these are factors that have been associated with treatment response. The most important is what type of hepatitis C you have. In North America, basically we're talking about genotype 1, genotype 2 and genotype 3 hepatitis C. Genotype 1 is harder to treat, genotype 2 and 3 respond much better. We know that patients, the level of the viral replication in the blood before we start treatment, that has an important effect on how well or how likely you are to respond to treatment. And there are various different thresholds between 400,000 and 800,000 international units per mil. We know that African Americans respond much more poorly to treatment than Caucasians. That had been a mystery for a long time, and some of the work we've been doing here is shedding light on part of the reason for that. And then we know adherence to therapy is important, and that's pretty self-explanatory. If you don't take your treatment, it's not going to work as well. And then there are a number of other things that have been weakly associated with treatment response, and whether they're significantly associated or not has varied from trial to trial. So the dose of the interferon, how high the dose is of the ribavirin. Females in most studies respond better than males, but not all studies. Younger patients tend to respond better. Patients who are not obese tend to respond better. Insulin resistance has recently been identified as a factor that's associated with poor response rates. Elevated ALTs, which is thought to sort of reflect how active your body's immune system is to clearing the virus. So the higher your ALT, the better you tend to do in the majority of studies, but again, not all. And so the idea there is that the interferon that we're treating with is piggybacking onto the active immune response of the body. And then cirrhotics are much harder. So cirrhosis really should be in the major predictors. So if you're cirrhotic, it's much harder to clear the virus. So this shows the difference in SVR rates between genotype 1 and genotype 2-3 patients. So if we think about that 50-55% overall success rate, for genotype 1 patients, that's much lower than for genotype 2 or 3 patients. So for genotype 1 patients, the success rate is 40 to 45%. For genotype 2-3, it's 75 to 85%. Recall that most of the patients in North America are genotype 1. Now the standard treatment regimen is a year, but in genotype 2-3 disease, because they respond so much better, they only need 24 weeks. You can see here that the success rate of treatment is exactly the same whether you're treated for 24 or 48 weeks in this large trial. So that's what the current standard recommended regimens are. For genotype 1, combination treatment, 48 weeks, 45% cure rate. For genotype 2-3, 24 weeks, 75 to 80% cure rate. Now in African Americans, who I was alluding to before, don't respond as well. For some reason, African Americans almost universally have genotype 1 disease, and it's not clear yet whether that's reflecting sort of how they got the infection and the pool of virus that they're exposed to, or whether it reflects a differential immune response within the African American population to the different viruses, and maybe they're just not susceptible. Maybe they clear hepatitis, the genotype 2-3. Maybe they're just not susceptible to infection with those viruses. But with the standard of care treatment regimen, so the 48 weeks, PEG interferon and ribavirin, only 20 to 25% are cured. 75% are not cured. The other thing in this population is that some of the other rules that we sort of apply, so cirrhosis, so the level of fibrosis, the level of viral load, adherence, a number of the host and viral factors don't seem to apply nearly as well in predicting which of the African American patients will respond. And then this slide just goes to the practical problems of treating hepatitis C as well as the issues of adherence. So despite me standing here and saying for genotype 1 disease we can cure half of all patients, this is a real world study where they looked at a pool in a clinic of 293 Hep C patients. Only 63% of them were eligible for treatment, largely related to the side effects of interferon. Of those 63%, only half accepted treatment or wanted to go on treatment. Of that 28%, only 22% completed treatment, and then 4% achieved an SVR. And this is slightly older data. We would expect the response rates to be better. So you can see that despite the 50% success rate, once you look at the entire pool of patients and we exclude patients who are not eligible, who are not interested in treatment, that 50% falls right down. Now, the big side effect of hepatitis C treatment, flu-like symptoms are the number one side effect. 70 to 80% of patients will complain of flu-like symptoms. The reason for this, so when we get the flu, the symptoms you feel are the body's interferon response to the flu virus. So we're giving mega doses of interferon as part of the treatment for hepatitis C, and not surprisingly you see flu-like symptoms. But there are lots of other side effects. So you can see almost 70% of patients, fatigue, headache, myalgia, rigors, fever. So the flu-like symptoms. But nausea is a common problem, insomnia, alopecia, arthralgia, irritability, depression, anorexia and pruritus. It's pretty, I would estimate to most patients they will probably lose 5 to 10 kilograms of weight on interferon treatment. And for patients who have a history of depression, I would put them on prophylactic antidepressants if they were happy to go on them. So this brings us to, this is more of the DCRI part of the talk.
Alex, before you go on, you talked a little bit about interferon. Do you want to talk for like 2 minutes about what ribavirin does?
No, not really. So we don't know a lot about how interferon works, but we really don't know anything about how ribavirin works. So John McHutchison was actually he ran the first big study using ribavirin, and the sort of background story is they ran this trial in combination with interferon on the basis of some cell culture models that showed that ribavirin had an antiviral effect against viruses completely unrelated to hepatitis C. And then there was a clinical study showing that ribavirin decreased the ALT levels in patients before we could routinely measure viral load. So it was thought, great, we'll combine the two. And then by the time they could combine the two, fortuitously it worked really well, they could measure viral loads in patients. So they looked back at the first ribavirin study where the ALT, the liver function tests, had improved a bit, had absolutely no effect on the viral load. So if they'd been able to measure the viral load in those patients, they would never have combined it with interferon. But for some reason, in combination with interferon, it almost doubles the response rate. So it has a number of different effects in vitro, some of which and we're not sure which is the most relevant to interferon. So it increases the mutation rate of the virus. As the virus is replicating, it increases the mutation rate. And the theory is that it causes less fit virus to be produced, which the interferon can then clear more easily. It can act as a chain terminator, so similar to some of the HIV and Hep B drugs, that as the DNA or the RNA of the virus is replicating, the ribavirin can be incorporated into the extending chain, and then because the ribavirin is there as opposed to a nucleotide or a nucleoside, the chain can't be extended, so it stops replication. Has effects on the immune system. It can boost T cell responses, but we don't know. But in view of all this toxicity, and in view of the fact that only 40 to 50% of patients respond to treatment, it would be really nice if we could predict which patients are going to respond to treatment so that we could channel our resources to treating those patients who are most likely to succeed, and we could avoid causing side effects to the patients who aren't going to be successfully treated. Now, the question we've been particularly interested in is there a host genetic explanation for this? There have been a couple of different cures. Just the fact that 50% of patients respond to treatment suggests that there's probably something in the host genetics that's causing a switch between responding to treatment and not responding to treatment. And then the other big thing is the difference in response rate between European or Caucasian patients and African American patients, suggesting that evolutionary there might be a difference that leads to a difference in response rate. And this is a project we've been working on this year. So we've done what's called a genome-wide association study to look at this. So a little bit of background. There are 23 chromosomes in the human body, and of those 23 chromosomes constitute over 3 billion nucleotides. So DNA consists of a helix of little building blocks or nucleotides, of which there are 4 attached end on end, one to the next. 99% of these nucleotides are identical between all of us. But you can see what are called polymorphisms. So in approximately 1% of the genome, or 10 to the 7 of these nucleotides, will vary from one person to the next. And this slide summarises a DNA helix, and you can see that these are 2 individuals. You can see that the sequence here is identical, but in this position here that's highlighted, you've got a C to G, which is a cytosine to a guanine nucleotide pair, and that's varied in the second patient to an A to a T. So there are over 10 million of these polymorphisms in the human genome. So the polymorphism, just the term single nucleotide polymorphism, refers to a switch of just one of these nucleotides, and it's a nucleotide variation that is inherited and is differed between individuals. And we think that, so your DNA code leads to phenotypes. It causes everything about us. So these polymorphisms have been linked to hair colour, they've been linked to height, they've been linked to various diseases and drug responses. So we were interested in hepatitis C. Now, as you can imagine, sequencing 3 billion nucleotides is something that technically is just becoming possible, but it's a very big endeavour and it's very expensive. So about 5 years ago, technology was developed that could use what are called tagged polymorphisms on chips to capture most of the variation in the human genome. Because most of these nucleotides are inherited together, you could use, say, this A to T mix here to capture the variation in sort of 10 or 20 nucleotides either side of it. Does that make sense? So instead of directly sequencing the entire sequence, we could pick out every 100, or it's approximately every 1,000 the way it works, but we could pick out every so often one SNP that would reflect variation within all the other adjoining SNPs, and this is due to what's called linkage disequilibrium. So you can use chips that have 600,000 of these polymorphisms that will cover about 90% of the entire variation across the human genome. And then the way these tests work is you run a statistical test for each of the polymorphisms to see whether it's associated with your phenotype of interest, which means that for a 610,000 marker SNP chip, you run 610,000 statistical tests. So multiple testing becomes a big issue. So that's what we've done in a population of hepatitis C patients, and this was published in Nature earlier in the year. So we did a genome-wide association study looking at genetic factors that predict treatment outcome or are associated with treatment outcome in genotype 1 patients. So to do this, John McCutcheon had run the IDEAL study, which was the first study, the first large-scale study to compare the 2 commercial preparations of interferon. So look, I haven't discussed this, but interferon alpha in the body, there are 2 companies that make a commercial preparation. There's interferon alpha 2A and interferon alpha 2B. Clinicians' impression has always been that they work pretty much identically, but no one had ever done a big study to compare them. So John ran that. It was actually an FDA-mandated study. So it was the FDA mandated that one of the companies do a big dose-finding study, and they decided to piggyback on their competitor's drug if they were going to be running this study anyway. And so John coordinated this study over 3,000 genotype 1 patients within North America, and what it confirmed is that the drugs work exactly the same. But it gave us the opportunity to do this fantastic genetic study because half of the 3,000 patients consented to genetic testing. So this is just the workflow for a genome-wide association study. So we had 1,604 patients from the IDEAL study, and then we had a further 67 patients from a study that Andrew Muir ran that was published in the New England Journal of Medicine in 2004. And his study had specifically looked at the difference in response rates between Caucasians and African Americans. We were particularly interested in that, so we wanted to increase the number of African American patients we had in the study. There were various exclusionary criteria based on complete data sets, based on quality control for the genotyping chip, which left us with 1,137 samples that could be validly incorporated in the analysis, and 550 or 565,000 polymorphisms for the association tests. And what we found was that a polymorphism upstream of the IL-28B or interferon lambda 3 gene was associated with an increase in SVR rate in adherent genotype 1 HCV patients. So this is called a Manhattan plot, and what it presents is the chromosomal position for every SNP that was tested on the chip, against on the y-axis the p-value for association. So you can see here that there are thousands of polymorphisms that have p-values for association nudging up to sort of 10 to the 4, 10 to the 5. You do this many tests, you're going to see lots of sort of apparently strong association signals, but once you adjust for multiple testing, they're no longer significant. And then when we get to chromosome 19, you see this skyrocketing of 7 SNPs with the top polymorphism RS 12, 97, 98, 60 having a p-value for association with SVR of 10 to the minus 28. So it's a strongly significant result. This polymorphism is very strongly associated with treatment response. And what does that translate to? What does that translate to? It translates to, depending on the variant you have at this polymorphic site, a doubling in your SVR rate. So there are 2 possible alleles at this polymorphism, a C or a T. And in patients who are CC at the allele, in Caucasians, in African Americans and in Hispanics, there was a doubling of SVR rate compared to the patients who didn't have the favourable genotype. Another interesting thing to come out of this study was that if you looked at African Americans who had the good genotype, they actually responded much better than the Caucasians who didn't have the good genotype, showing that in this instance it's this genetic marker, it's not the sort of your ethnic background that's determining your treatment response rate. But the frequency of the good allele, or the good genotype, is much more common in Caucasians than African Americans. So in Caucasians, the CC in dark blue, which is the good response genotype, was present in 39% of the population, compared to only 16% of African Americans. And we were then able to extrapolate to other ethnic populations. And you can see that the frequency of the C allele in the population almost exactly parallels the SVR rates that we see that have been published in the literature for SVR rates. So African Americans have a low frequency of the C allele, have a very low percentage SVR rate. Hispanics, the C allele is less common than Caucasians, and again SVR rates are lower than in Caucasians. And then in patients of Asian background, almost everyone is CC, and they have a very high response rate, higher than Caucasians. And by logistic regression modelling, it was estimated that the differences in our study of the frequency of the good genotype between Caucasians and African Americans explained half of the difference in their response rate. So it didn't explain everything, but it explained half. And one other thing that's been done is that population sampling has been done around the world that mirrors these results. So the bright green presents the frequency of the C allele in different populations around the world. So you can see in African populations, the good genotype the frequency of the good genotype is very low. It's a bit higher in North America. It's even higher in Europe, which sort of, for a long time we've known that European trials, the SVR rate's always about 10% higher than North American trials, and it have been wondered whether this is due to obesity or other factors. This is almost certainly the explanation. And then out across to Asia, where almost everyone has the good response genotype, and the patients again respond much better. So we don't know exactly how this is working yet, although we're working hard on it. But one of the things we do know is that it profoundly affects your interferon sensitivity. So this slide presents the viral kinetics on treatment for patients who are TT and CT, which is the bad response genotypes, versus CC, which is the good response genotypes. And the week numbers haven't come through. But on the left, this is week 2, week 4, and week 12 of treatment with interferon and ribavirin. So you can see that by week 2, the viral load reduction in the good responder genotypes is almost 3 logs, and it's more than 2 logs greater than the poor response genotype. So this is a really dramatic difference.
Can I ask a question?
Yeah.
So, back here.
Yep.
So with the SNP here in this reading frame, are we changing the amino acid, or do we know?
No. So the top discovery SNP is actually upstream of this IL-28B gene.
Okay.
Because these SNPs use the approach used is that they tag the region. So the sort of discovery SNP in most cases is not the polymorphism that's actually having the biological effect. It's just tagging a region within which there'll be an important polymorphism. So the discovery SNP itself is in between the genes and doesn't seem to do anything. But it's very strongly associated with a missense mutation within exon 2 of the IL-28B or interferon lambda gene.
So, I mean, it could have to do with just the whole histone pulling apart for transcription translation of other proteins.
Yeah, look, it's possible. Our best guess at the moment is that this is affecting the IL-28B protein itself. We think that this polymorphism within the open reading frame is going to be the causal SNP. And they're sort of in this population of 1,000 patients, everyone who had the sort of good discovery polymorphism on the chip, when we sequenced them, also had the sort of favourable polymorphism in the open reading frame. And we've looked at expression. It doesn't seem to affect expression of the IL-28B. We think it probably affects receptor binding, and that's going to be the mechanism, although we haven't shown that yet.
So some amino acid change or some sort of folding.
Yeah, so there's an amino acid change. Yeah, there's a lysine
right.
to an arginine change within exon 2 of the open reading frame.
And that definitely that particular switch would definitely affect your folding and conformation. I mean, depending upon the location.
Well, probably, yeah. So the protein itself was crystallized earlier this year, and they did a whole lot of mutagenesis studies. They looked at almost every residue except the one that's affected by our sort of hypothesized causal variant. But the residues either side of this one, there was an effect on receptor binding. So it's not proof, but we think that's how we think it's working.
Thanks.
And it's not just us. Hot on our heels, within 4 weeks, there were 2 independent genome-wide association studies that essentially showed the same thing. And then the other interesting thing is that this polymorphism, beyond being associated with treatment response, has also now been shown to be strongly associated with spontaneous clearance. So when I spoke about 20 to 30% of patients clearing the virus when they're first exposed to it, it seems that the likelihood of doing that is 2 to 3-fold higher if you have this good responder genotype. So the way I think about it is that this polymorphism is regulating interferon alpha response, whether that's endogenous, as would be the case in spontaneous clearance, or whether it's treatment-related and pegylated interferon. But I think it sort of adds weight to the sort of biological significance of this, that it's not just related to treatment response, it's also related to spontaneous clearance. So to conclude this sort of genetic association study, in genotype 1 patients, the IL-28B genotype is strongly associated with increased SVR rate. It profoundly influences viral kinetics. It explains much of the ethnic differences in response rates. And I didn't present this data, but it's the strongest pre-treatment predictor of SVR in these patients, so much stronger than fibrosis or viral load or other factors that have been associated with treatment response. So where are we going to take this? First of all, we think this will be clinically applicable as a diagnostic. So since these results have been published, we've been receiving calls from all over America about patients wanting to have this test done. You can imagine that if you're a patient with genotype 1 disease and your SVR rate was estimated at 50%, if you could have this test done and therefore differentiate yourself into an 80% responder or a 30% responder, that'd help you make the decision. Not everyone has genotype 1 HCV, and we don't know how relevant this polymorphism is to the other genotypes, although we're collaborating with a group in Italy. We're running that analysis at the moment. Maybe in the good responder genotypes, they don't need 48 weeks of treatment. Maybe they only need 24 weeks of treatment, like the genotype 2-3 patients. That would be attractive. It would save significantly in the costs, and it would also avoid an extra 24 weeks of side effects. And then the treatment paradigm for these genotype 1 patients is about to change, largely due to studies that have been run through the DCRI with the introduction of direct antivirals for hepatitis C. So we don't know whether this polymorphism, whilst it's very relevant to the current standard of care, we don't know whether it's going to be relevant to the new therapies that are going to be available in a couple of years. So that brings us to the last part of the talk, which is a short overview of the direct antivirals. So these direct antivirals, similar to drugs that are used for hepatitis B and HIV, directly attack steps within the life cycle of the hepatitis B. They're referred to as the Stat C drugs within the sort of hepatitis C world. So specifically targeted antiviral therapy for hepatitis C. This is the reason I put this slide up earlier. So the major classes of these drugs, first of all, the NS3-4A protease inhibitors affect the translation and polyprotein processing necessary for viral replication. That's due to this NS3-4A protease. The second class of drugs are the NS5B polymerase inhibitors that directly inhibit reverse transcription and replication. And then the third class of direct antivirals coming through are the NS5A inhibitors. So the NS3-4A, NS5A and NS5B are all proteins that the hepatitis C makes. We know pretty much what the NS3-4A protease does and the NS5 polymerase does. We don't know exactly what the NS5A protein does, but we know that inhibitors of it inhibit viral replication. So this slide summarises the different drugs that are in phase 2-3 development at the moment. Now, all of these drugs are being used in combination with pegylated interferon and ribavirin. So these aren't going to be a magic standalone pill. When they come to market, they're going to be combined with the current standard of care. The top two candidates at the moment are telaprevir, which is the Vertex drug, and the phase 2 program was run through DCRI, and boceprevir, which is a sharing drug. And I'm going to limit my discussion, and you can see that there's 10 or 12 on the list here, and then if you include preclinical and phase 1 trials, then there's over 50 or 60 drugs in development at the moment. But telaprevir and boceprevir will be the first that come to market. So this PROVE-1 on the left was the study run through DCRI, and this used telaprevir, which is the protease inhibitor, and it compared standard of care. So all these drugs to date are genotype 1 specific, so they don't work for the other genotypes. So you can see that in the control arm, treated with the standard of care, PR means pegylated interferon and ribavirin, for 48 weeks, the SVR rate was 41%, which is about what we'd expect from all the previous literature. And then 12 weeks of telaprevir was combined with 3 different regimens of the pegylated interferon and ribavirin. So 12 weeks of telaprevir with 12 weeks of peg ribavirin. This was an FDA-mandated arm, and then they got gun-shy and decided it wasn't such a good idea. So there are only 17 patients, but they didn't respond very well. But once you got telaprevir in combination with 24 or with 48 weeks of peg ribavirin, you can see that the SVR rate's almost doubled. So 61% and 67% of the patients treated respectively for 24 or 48 weeks were cured of their hepatitis C, compared to 41%. And then in PROVE-2, which is a European study of a very similar design, the SVR rates were 60% and 69%. Note here, this is really pretty exciting, that in their 82 patients who were only treated for 12 weeks, the SVR rate was 60%. So these drugs work better, and it may be that we can shorten treatment. So we may be able to get away with 24 or even 12 weeks of treatment. And it's not just in treatment-naive patients. So the pool of patients that we see at Duke now, we see an awful lot of patients who've been treated with pegylated interferon and ribavirin. It hasn't worked, and then they sort of hang around waiting for the next thing to come through. And as a general rule, if you failed treatment with one regimen, it doesn't work as well the next time. So you can see that in patients who'd previously failed treatment, if you retreated them with standard of care, only 14% responded. But if you added in telaprevir for 12 weeks or for 24 weeks, the SVR rates were 51 and 53%. So the same as in treatment-naive patients. And this is really a big step forwards, because within clinics, this is becoming a much bigger pool of our patients. Once patients are cured, we send them on their way, and we just progressively accumulate this larger and larger cohort of patients who haven't responded to treatment. And then if you divide patients up into sort of how badly they responded, so non-responders are patients who have been treated with interferon and just never cleared virus from their blood. They were positive all the way through. They don't respond as well, but for this very difficult group of patients to treat, 40% still responded. And then if you looked at patients who responded on treatment but then relapsed after treatment, the response rates were 70%. Now these are phase 2 trials, and the phase 3 trials are underway. They'll need confirmation in bigger numbers, and the numbers may change a little bit, but it's very encouraging data. And then the competitor is boceprevir. Now this was a horribly complicated study where they actually had 6 arms, but for the 5 arms they had a standard of care treatment for 48 weeks. Then they had boceprevir in combination with standard of care for 28 or 48 weeks. and then they had two other arms where they treated with pegylated interferon and ribavirin for 4 weeks before starting another 24 or 44 weeks of treatment with the boceprevir in combination with interferon and ribavirin. And you can see that this is a protease inhibitor that acts, it's a different drug but acts at the same target, and the response rates again were pretty similar and very high. Now, as with all of these drugs, the reason they had that 4-week lead-in was they wanted to make sure they had high levels of interferon before the direct antiviral started because of the spectre of antiviral resistance. So again, similar to HIV or hepatitis B, if you use these direct antiviral agents, viruses mutate, that's what they do, that's how they survive. And in the setting of monotherapy with a single direct antiviral, resistance is almost inevitable. And so you can see here, this is why the drugs are going to be used in combination with interferon and ribavirin. So in the telaprevir and boceprevir studies, in combination with PEG and ribavirin, breakthrough due to viral resistance was less than 10%. One of the key ways ribavirin is working in this setting is stopping resistance. So if you had telaprevir just in combination with interferon, it was better than telaprevir alone, but the resistance rate was almost 25%. So it's 1 o'clock, I'm going to wrap up. So the burden of hepatitis C is increasing at an alarming rate. Improving SVR rates and improving the number of patients treated may help to reduce the burden. The IL-28B genotyping is new, but it's likely to have important implications for treatment decisions. And we also hope that it's going to bring more patients. So the way the drug companies have been thinking about this is, well, why would we want to do this test? Because for over half of patients with the bad genotype, they're not going to respond well. It's going to decrease our market share. But we would hope that this would actually bring more patients forwards, and that it becomes more attractive if you can have a test to see whether you're a good responder, and you're going to attract more patients who have the good responder for treatment. And then the directly acting antiviral agents currently completing phase 2 combination trials can achieve both higher SVR rates and shorter treatment durations in treatment naive and treatment experienced patients. And again, we hope this will encourage more patients to initiate and complete therapy. And then finally, I'd just like to acknowledge everyone that I've been working with here at Duke. And that's it. Thank you.
Thank you very much, Alex. I know people probably have lots of questions, and Alex has kindly consented to stay around for a little bit longer and answer some of those questions.
So this last set of slides you showed, do they have less side effects, or are you just saying that?
Sorry?
The last set of slides you showed, is the side effect still the same? Why would they be more likely to come, other than to know that either they will be responders or not?
I think for a patient, the idea that I can go through this 48 weeks of treatment with toxicity for a 50% success rate puts a lot of patients off. So if you're a patient with hepatitis C who's otherwise pretty healthy, you don't feel sick, why would you put yourself through that if the likelihood of responding is only 50%? I think if the response rate becomes 80% and we can even get away with 24 weeks of treatment, that just becomes much more attractive. So, and so we have no data that, it's possible that in these patients, side effects, they might even be greater. We haven't done that analysis.
We have the protease inhibitors for once the virus is in the cell, but what do we know about the entry into the hepatocyte?
For the protease inhibitors?
No, the entry of the virus.
So,
they've developed HIV. Sorry.
So it would be nice if we could have an entry inhibitor. We don't know what the receptor for hepatitis C is.
So we don't know the entry mechanism.
Well, it's thought to be receptor mediated, although we don't know what the receptor is. Yeah. Does that make sense? So Claudin-1, the LDL receptor, and SRB1 have been hypothesised to be the receptors, and the sort of in vitro models that have supportive evidence for all three of them. But we're not sure exactly which one it is.
I'll ask you one more difficult question, Alex. So for hepatitis B, some of us remember before there was vaccine for Hep B. Is there a reason why for Hep C, is it that there will not be a vaccine because there don't seem to be, are we heading in that direction?
So
even for HIV, of course, a lot of people have tried to produce vaccines unsuccessfully so far.
Yeah. Look, it's probably related to the mutation rate. We're lucky with hepatitis B that there's a couple of different things about hepatitis B compared to hepatitis C. Hepatitis B has overlapping reading frames. So whilst it can mutate not quite as much as Hep C, mutations affecting the surface, so that the polymerase overlaps the surface reading frame, so the vaccine is targeted against the surface protein. And so mutations that will mutate away from the vaccine will have a cost to the polymerase. So it's much harder for hepatitis B to mutate away to escape from a vaccine. And humoral immunity seems to be more important. With hepatitis C, it mutates a lot more. It seems to be able to evade, certainly in the setting of therapeutic vaccines, it seems to be able to evade any immune response that is boosted by the vaccine. Mm-hmm. I'm pessimistic. I mean, things may change, but with the current vaccine approaches, I'm sort of pessimistic that we're going to see anything soon.
Other questions? Everyone's very quiet today, so I'm assuming that this means you have absolutely covered this territory fabulously well, because this group usually at these sessions asks a lot of questions. And actually, when you were talking, I was thinking this is probably the best, most comprehensive presentation I've heard in a long time on any of the infectious diseases, but certainly on hepatitis. So really, on behalf of everybody, thanks so much, Alex. And really, I think congratulations in terms of being involved with really cutting-edge research. So
it's been really exciting.
Yeah, so we definitely appreciate actually having one of the co-authors of this. Really, I think it's a seminal study in terms of. So it's kind of fun to be at the DCRI at Duke. For those of us who complain intermittently, like maybe once a day, about the DCRI, I think that we have to look at, this is the reason that we're at the DCRI and not at Quintiles, because we get to interact with people like Alex and really hear presentations that are so cutting edge. And I know that you presented at the liver meeting at AASLD as well. So really, thanks, Alex, and really appreciate having one of the people who are working on hepatitis C really at the forefront of research. So thanks very much.
No problem.